Thanks very much, Linda. Hello, everyone. Nice to see everybody's faces. So I'm Dr. Jordan Seeley. I'm a molecular microbiologist / genetic epidemiologist, and I'm based at Bangor University at the moment. And then I'll hand it over to Jack so he can introduce himself.
I'm Jack. I'm a final year PhD student based at the University of Aberdeen.
Okay, Brill. So without further ado, we'll go straight into our next lot of speakers. So this topic is on emerging technologies and new frontiers in AMR research. So we've got our keynote speaker, Professor Chris Creevey from Queen's University, Belfast, and he'll be talking about mutually exclusive genomic constraints define the limits of antimicrobial resistance evolution. So we'll welcome Chris.
Hi, first of all, thanks very much to for the organisers for the invitation to speak today and talk about some of the work we've been doing in the area of AMR and machine learning. And I say we in a very loose context. Lucy, who's down the bottom of the screen here, is a postdoc in my group who's been pretty much pushing all of the work that I'm going to talk about today. And she's there in the back if you want to talk to her about it later on. I'm going to talk a little bit about the work we've been looking at. Everything I'm going to talk about is all computational-based analysis. We're a computational biology group based here in Queen's, just up the road in the School of Biological Sciences, and we work in the area of microbiomes and genetics and trying to understand how systems work. So we'll get into it now. So I don't really need to tell this audience about the global threat of AMR and the problems that it poses in the future. But one of the things I do want to just highlight is when we talk about AMR from a genomic context, and we talk about how resistance is acquired, we tend to think about the idea of a modular approach to acquiring resistance. You get a gene, you get resistance. You gather another gene, you get another resistance. And that's the idea that we have primarily driven through, if you're looking at the genomic context and you're analyzing genomes, that's the paradigm that we work in. And this is like a Lego block. I like to kind of think about it like Lego blocks. You know, you just gather more of these blocks and you can gather as many different resistance as are necessary. So I want today in this talk to challenge that idea and bring you through a kind of a story of the analyses and the ideas that we've been exploring that perhaps tell a different story to that paradigm. So we'll just start with the simplest question. If you have an AMR gene in a genome, does that equal a resistance phenotype in the organism? And this is work where we did a very simple thing, gathered about 6,000 genomes from across 19 bacterial genera, and those genomes had data from UCAST-linked MIC values for across 23 antibiotics. So we had the genome and we had the experimental evidence of what they were resistant to. And on the left there is a graph which shows the 23 different antibiotics and how accurate it was to ask the question, I have this AMR gene, does that equal resistance? And on average across all of the antibiotic resistance genes, it was about 56% accurate. So it's just a little bit better than flipping a coin. And there were some that were particularly bad, like clindamycin was particularly bad, but that's a data issue. We have very little data on that particular one, so predictions of that are not great. And some are really good like moxifloxacin. So this shows that the idea that AMR gene presence equals resistance is flawed. And so we went from that point onwards to try and investigate what we could learn from that. So we asked the question whether or not, well, if it's not just one gene and present that is related to resistance? What if we started to expand the search in the genome and start to look at questions about whether it's combinations of genes? Maybe we should look across all known AMR genes and ask, well, maybe even though this AMR gene is known to convey resistance to a different antibiotic, maybe somehow it's also involved in the phenotype of this other antibiotic resistance as well. So you look across all of those genes. Or maybe if we expand it to the entire genome and we look at all genes in the genome and ask questions about whether or not we can piece together what genes might be related to resistance, what would we see? And so we applied some rather simple machine learning approaches to this. In particular, we utilized decision trees in this analysis because it allows us to go in and look at specifically what genes are driving the specific resistances. So on the left there, you can kind of see that we have the original graph I showed you in the yellow showing our accuracy, and then for each of the other graphs we started to look at different types of data or models applied to exactly the same problem and their accuracy of predicting the resistance phenotype. And as you can see, that increased quite significantly. And so it shows that if you start to bring in more of the genomic context of the genomes, you can get better predictions from the genome based on their accuracy. And I'm just going to briefly explain how this idea on the left, because I'm going to talk about it a bit, how these decision trees work. When we generate a model like this, we will get a model for a specific antibiotic, like up here on the right, and you start at the top, and you say, okay, in my genome, do I have gene A? Yes, okay, then I'm going to go down to the right. Do I have gene D? Yes. Do I have gene E? Yes. It's predicted to be resistant. So you follow it in a route like this. So what you end up with in a decision tree are what we call routes to resistance. You can end up with different combinations of genes that can result in different paths to a resistance phenotype. So I'm going to talk about that. So it shows, as I said here, that you need the right supporting cast, not just the star player, to have your resistance. Okay, so here's an example for a tetracycline model from our data set. So at the top here, they're listed with the COG names from eggnog gene families as a general guide. And everything that's bold here with a dark ring is a known AMR gene that has been discovered. Now, we didn't tell the models that these were known AMR genes. They just came out in the models as you would expect. But interestingly, if you follow these lines down to where you reach the red boxes, every red box represents a predicted outcome where we would predict that this genome is resistant. And interestingly, if you follow these different paths down, they're all numbered that correspond down here, and you look across all the genomes, you will see that generally you end up with species-specific routes. So these routes 2 to 8 are primarily Neisseria genes, Campylobacter and Streptococcus. But this is all in all the genes and all the routes towards resistance to the same antibiotic. So we see species-specific routes to antibiotic resistance. So immediately we know you can't just take a general rule for every species out there. So this taxonomic structure isn't random. We think this is fundamental to what it is to be to the resistance. Okay, so what if we look within one organism? Would we see similar patterns within one organism? We'll take E. coli, for instance, and we look at generating models for resistance within E. coli, but in this case, we take multi-drug resistance. And we ask the question, well, for every route to multi-drug resistance that we can reconstruct here, there is a question that gets posed. So, first of all, do we see any routes? What would you expect is, if we looked at all the pangenomes and all the genomes of E. coli, we would expect that all of these genes which represent a route to resistance should co-associate with each other in a pangenome. We should see them together all the time. But there also might be the case where certain routes may conflict with each other. So we examined this. So what we did was we put together 55 multi-drug resistance models, again looking at decision trees. And what we found was, as expected, we found when you look at the gene pairs in the roots to resistance, and you look at the pan-genome and you ask whether those genes are statistically associated with each other, so they're present all the time together across the pan-genome, we find 282 of these pairs match exactly what we found in our models. But interestingly, there were cases where we found disassociated pairs. So what does this mean? This means that there is genes involved in certain routes to resistance which are never found with the genes that are involved in another route to the same resistance in E. coli. So there are two independent routes to the resistance in E. coli, but you could only have one. You never see the two together. So these are mutually exclusive. And this is the box here in red, is where we found here in the box we have the resistance phenotypes of a dual resistance model for two different antibiotics. And we found that for the same, for the different types of phenotypes, whether you're resistant to both of the antibiotics or just one of them, there were gene pairs that were resistant to each other. So an example is the two genes up here, COG3316 and 3677. That's an example of two genes which are both transposases that are predicted to be involved in resistance and in the same resistances, but we never see them coexisting in genes. So we see you can have one, but not the other. So this suggests that alternative evolutionary routes to the same MDR phenotype exists, but some of these routes cannot exist in the same genome. Okay, so we can ask the question, well, if that's true in E. coli, is it only in E. coli? So let's take all of the genes that we've identified that are either statistically associated or disassociated with each other, involved in resistances, and let's compare them to the same genes in Pseudomonas aeruginosa. And we were able to identify 69 shared gene pairs that we knew to be involved in resistance in E. coli, and they were statistically associated or disassociated with each other in E. coli. And we examined the same thing by gathering a data set of about 8,000 Pseudomonas aeruginosa genomes and doing a pan-genome analysis as well. And this is the kind of thing we can see. Let me see, I have a, yes I do. So here is an example. Up here are all the associations or dissociations in E. coli, and down here are all those in Pseudomonas aeruginosa. Here's an example of just the same two pairs of genes, one in E. coli and one in Pseudomonas aeruginosa. In E. coli, these two genes are disassociated with each other. You cannot have them together in the same genome. But in Pseudomonas aeruginosa, they're statistically associated. So you see them together statistically quite often. So what we're seeing here is not really about like geography or environment. There's fundamental genomic incompatibilities here. There's the genomic grammar, whereby if you bring a gene into one organism, it can have a different effect if you bring it into another. And it's based on what's in that genome. So the same genetic combination is enabling resistance in one species is forbidden in another. Okay, so why can't bacteria have everything? So what are the possible mechanisms driving this? We could have resource competition, where you have multiple things like transposases competing for cellular machinery. You can only have one or the other. You can have membrane constraints. If we're talking about efflux pumps, you can only have so many of these in your cell membranes. There could be metabolic burdens in maintaining expression and replicating. You could have regulatory conflicts. You know, if you have different regulatory elements regulating the expression of these genes, they may conflict completely. And you might have an issue with genome stability, where excessive mobile elements compromise the structural integrity of the chromosome as a whole. So what this suggests, and these are all our hypotheses right now, the ways in which maybe this has been driven. But the underlying message is that evolution of resistance, in this case, isn't a limitless playground. You know, there are boundaries which are set by the organism's existing genomic architecture, and you cannot have everything. If we take a step back and we say, okay, well, that's one aspect of it. What about all the things that are shared? How easy is it to share genes and share different resistances? Well, this is an analysis where we took all of our models and we asked the question for every gene, how many times do we see any one gene in different resistance models for different antibiotics? And the dots here, the circles here are represented in how many models any one gene has been found. So that's a gene that's been found in a lot of models. And then we drew lines between them if they had two particular, had to be two criteria. One, those two genes had to be in the same model for an antibiotic resistance. And two, they had to have a predicted protein-protein interaction from, we use string to predict this. So we're looking for things that are predicted to interact, and we found them in the same model. This is the graph we see. It's a lovely diagram that's colored by the antibiotic resistance models it came from. But what you see is, first of all, nice structuring of independent genes that are only found in some antibiotic resistance models, but there are some key genes which possibly could be stepping stones towards resistance from one type of antibiotic to another. You get one of these, then you already have in place one or two genes that are important to resistance to another. You only have to get a few more to be able to get resistance to another sort of antibiotic. So in this case, you know, we expect that this might help us understand where MDR will evolve and where it's constrained overall. So we're back to the paradigms. The old paradigm then was that AMR genes are like Lego blocks, and with unlimited combinations, build what you like. But what we're suggesting is the paradigm we should be looking at is that genomic architecture creates these boundaries and constrains evolutionary trajectories of where you can go. And this suggests that the evolutionary landscape of resistance is predictable. You know, we've got all of these bacteria trying to get to the maximum, resistance, but they're all starting from different positions, which is defined by their genomic context, and they are restricted by that. And some routes are blocked because of genomic incompatibilities. And this suggests that evolutionary resistance is more predictable than we thought. So what are the possible clinical and surveillance implications? And again, these are our ideas where we'd like to go next. You know, recommendations on this is that you cannot do general models. You have to have species-specific models for this kind of work. There's a possibility of exploiting mutually exclusive pathways therapeutically. If you were to create a series of therapies, you could push microorganisms down a specific dead-end route. You know, we can monitor these hub genes for possible indicators of where routes of risks of horizontal transfer may exist. And we have the possibility of having this rapid computational screening using these computational models to do that work. So what we would like to do, if we get the chance to do anything like this, of course, this is all computationally predicted. We would like to go in and see whether or not our predictions are real. We would like to see if you knock in or knock out any of these genes, does this affect the phenotypes as predicted? Our frameworks are based on the organisms which we have available for, which are not always the high priority organisms. So we need to move our models over towards these more high priority organisms, but data is an issue. So we need data. If you have data, tell us. And we need mechanistic studies. Why are certain incompatibilities there? Is it because regulatory, metabolic, structural? And we think we need to develop clinical decision support tools, and we think that computational approaches on genomic data can help in that area. Two of the papers that are supporting this work, one in Life Science Alliance and the other on bioRxiv, are here. Lucy's in the back as well, and I'd be more than happy to talk to anybody who'd be interested in talking about that. And just to say finally, this all happens because of everybody in Queen's and my group and friends, and all of the support we get from our various funders. So that's it. Thank you very much.
Thank you for that great talk.